Hello. Hi.
Welcome to our vlog. Everyone, this is Group 5.
Hey,
Hi.
Hello everyone. This is Group 5.
Group 5, my name.
this is Group 5,
Welcome to Group 5.
Welcome to Group 5, and this is Gene Therapy. Gene Therapy.
the gene therapy.
This is Gene Therapy.
This is gene therapy.
Welcome to my vlog. Hello guys, welcome to my vlog. So I am a member of Group 5. My name is Elora Bastiero, BS Architecture 2C. And today I'm gonna discuss with you what is gene therapy and what is cell therapy and what are the differences between the both of them. Let's begin with gene therapy. Gene therapy involves the transfer of genetic material, usually in a carrier or vector, and the uptake of the gene into the appropriate cells of the body. Gene therapy replaces a faulty gene or adds a new gene in an attempt to cure disease or improve your body's ability to fight disease. So now that you've heard what is gene therapy, let's discuss the next, which is cell therapy. Cell therapy, aka cellular therapy, cell transplantation or cytotherapy, is a therapy in which viable cells are injected, grafted, or implanted into a patient in order to effectuate a medicinal effect. So let's talk about the difference between the gene therapy and the cell therapy. So first, let's go with the gene therapy, wherein they use a DNA to manipulate a patient's cell to cure a disease that it might be inherited or acquired. Meanwhile, the cell therapy, they use transplant within the whole cell to cure the disease. So let's see, there is a difference between the manipulation of cells and the transplant of cells. Now you know the difference.
First, I'm going to explain what is gene before yung gene therapy po natin. And gene is a basic functional and physical unit of heredity, and genes are made up of DNA. And short for, pwede natin ito mamana sa ating parents or sa lolo and lolas natin, sa ancestor po natin. And yung mga pwede po natin mamana sa kanila is yung physical appearance po natin, yung mga personality po natin, some other diseases po. And some genes act as instruction to make molecules called protein. Pero hindi po lahat ng ating genes is nakaka-produce ng protein po. Kaya po tayo nagkakaroon ng defective diseases. And dito po pumapasok si gene therapy po. And yung gene therapy is an experimental technique that uses genes to treat or prevent diseases. And yung technique po na ito is ginagamitan po siya ng vector na nagki-carry po ng gene na yung gene po na yun is nakukuha natin sa relatives ng patient na gusto nating gamutin. And yun po, ini-insert po yun dun sa patient para ma-treat po yung disease na nagkaroon po siya. So, yun po yung gene therapy. And dun po nag-e-end yung yung topic ko po.
So we will talk about gene therapy, the strategies of gene therapy. There are 4 strategies of gene therapy. They are gene augmentation therapy, targeted killing of specific cells, targeted mutation correction, and targeted inhibition of gene expression. So the first one is gene augmentation therapy. So what is the gene augmentation therapy? According to my research, it is used to treat diseases caused by a mutation that stops a gene from producing a functioning product, such as a protein. So, gene augmentation therapy, ito yung method kung saan yung gene transcript ng isang DNA ay tinatama na especially those genes na may disorder o hindi tamang pagkakalagay ng GTCA protein sa DNA ng strand. Ito ay para maging tama ang isang gene at mag-function ng gene sa tama nitong gamit. So, number 2 strategy of gene therapy is targeted killing of specific cells. So, this aims to insert DNA into a diseased cell that causes the cell to die. Genes are directed to the target cells and then expressed so as to cause cell killing. So it can be done by two ways. Number 1 is direct killing and number 2 is indirect killing. So what is direct killing? Direct killing is the inserted DNA contains a suicide gene that produces a highly toxic product which kills diseased cells. And indirect killing uses immune stimulatory genes to grow or enhance an immune response against the target cell. So, sa pangatlo nating strategy, ito yung targeted mutation correction. Sa research ko, ang targeted mutation correction is the repair of a genetic defect to restore a functional allele. Technical difficulties have meant that it is not sufficiently reliable to warrant clinical trials. In principle, it can be done at different levels, at the gene level or at the RNA transcript level. Sa pangatlo nating strategy, ito naman ay tinatama niya yung mga may maling transcript sa RNA upang maging ganap na functioning allele. Dito mo malaman ang blood type o ang status ng chromosome. Dito, inaayos niya yung may mga abnormalities para maibalik sa normal at order ang isang gene. So, lastly, ang pang-apat nating strategy is targeted inhibition of gene expression. So, targeted inhibition of gene expression. This is suitable for treating infectious diseases and some cancers. The basis of this therapy is to eliminate activity of a gene that encourages the growth of diseases-related cells. If disease cells display an inappropriate expression of a gene, a variety of different systems can be used, specifically to block the expression of a single gene at the DNA, RNA, or protein levels.
So on the previous video clips, we have discussed some important details about gene therapy. Now I am going to discuss about the uses of gene therapy and why is it done. Okay, so just a review guys, gene therapy is used to correct defective genes in order to cure a disease or help your body better fight disease. So some researchers like Mayo Clinic are investigating several ways to do this, including replacing mutated genes. Some cells become diseased because certain genes work incorrectly or no longer work at all. Replacing the defective genes may help treat certain diseases. The next one, fixing mutated genes. Mutated genes that cause disease could be turned off so that they no longer promote disease. Or healthy genes that help prevent disease could be turned on so that they could inhibit the disease. So the third one, making diseased cells more evident to the immune system. In some cases, your immune system doesn't attack diseased cells because it doesn't recognize them as intruders. Doctors could use gene therapy to train your immune system to recognize the cells that are a threat. So aside from the 3 uses that I have mentioned, there are 6 more uses of gene therapy. First one, it replaces missing or defective genes. hastens the speed of destruction of cancer cells, supply genes that cause cancer cells to revert back to normal cells, delivers bacterial or viral genes as a form of vaccination, provide genes that promote or impede the growth of new tissues, and the last one delivers genes that stimulates the healing of damaged tissue.
The difference between gene therapy and cell therapy is, in the broadest sense, removal or change in the content of a person's genetic code with the goal of treating or curing a disease. That is gene therapy, while cell therapy is the transfer of intact live cells into a patient to help lessen or cure a disease. That is cell therapy. And the most common type of cell therapy is blood transfusion, the transfusion of red blood cells or white blood cells, and the platelets from a donor. That is the most common type of cell therapy.
So one way scientists introduce new genes into the body is through the use of vectors. In this case, viruses. Scientists will take these viruses, in this case vectors, and then erase the harmful effects that it has. And then they will introduce new genes into that virus. Then they will use that virus as a vehicle to introduce these new genes into the cell. As the virus enters the cells, these cells will then eat away on this virus, leaving the gene to enter the body. That's how scientists introduce new genes into the cell. So scientists use viruses depending on which cell they are targeting. So if they're targeting liver cells, they will use a liver virus as a vector. Now you might be thinking, since we are using viruses as vectors, as vehicles for these good genes, wouldn't that have harmful effects to our bodies? In this case, it depends. Sometimes these viruses target multiple cells in our bodies, and that will cause complications in the process. But with that said, scientists and doctors are making sure that these risk factors don't actually happen. So when it comes to gene therapy and cell therapy, it is something we can rely on. So gene and cell therapy is all about trying to solve medical problems that we are experiencing in this very day but we need to keep in mind that there are risk factors to this gene and cell therapy. So my name is Carl.
I'm Maria Kristina Ballogdajan.
This is Francesca Diaz.
This is Gerard, or you can call me G.
I'm Elora Bastiero.
I am Diaz, and this is Group 5, and this is Gene Therapy.
This is Group 5.
The gene therapy.
And this is gene therapy.
Presenting and saying goodbye.